All right. All right, so of course we're here today to talk about Patterns Biology Unit 2. Just a couple of additional documents that we will not be looking at in detail today, but that could be helpful for you as you kind of try to get your mind around all of the different things in this unit. The first one there is the Unit 2 outline. That gives an overview of all of the tasks in the unit. It has links to all of the tasks. And it allows you to kind of see the whole thing on one document. I think it's like 3 or 4 pages. It can be a little bit intimidating. That's not our intention. The intention of providing that outline is for you to do exactly that, to be able to see the whole thing in its entirety, along with extensions and assessments. It provides details about the standards that are covered in each one. Let me just shift this over here just for a second to show you. It provides some rubrics, although most of the rubrics are embedded in each additional task or individual task. It tells you the all of the 3 dimensions for each task and so on. So this is not something that we're going to be going over today, because we are going to be looking at individual tasks and individual types of documents within this, but this is for you to refer back to later. The second document that I have linked here is the student interactive notebook for this unit. There is also on the unit outline a student interactive notebook, a teacher version that was done by my colleagues at Sunset High School in Beaverton. That one we don't have access to share with you to share with you a student version, but you can see what the teachers did with that if you prefer to try a slides version. I've kind of gone back and forth and always come back to Google Docs. I find that it's the easiest for students to handle. So I'll refer to that. I like to call that the INB, Interactive Notebook, INB. So if I refer to the INB, that's what I'm talking about. This is something that in my classroom, the students are interacting with that every single day. My colleague next door uses a physical paper notebook. She uses all of the same prompts, but decides to do it in a paper version. So kind of gives you an idea about what the type of things that we're asking students to do on a daily basis for note-taking. All right, any questions before we get started? I don't know what happened to there's my chat. I had to get my chat up on my screen here. All right, so in Unit 2, we're asking the big question about how molecules are used to build cells. Our Unit 3, it's kind of bookended on one end. This is the first experience that students are coming in looking at molecules with. The previous unit, of course, is about ecosystems and biodiversity. So we do a big jump from macro to micro right away in October. And then on the other end, we have a unit that we call homeostasis, or cells to organisms, and it takes the basics of how cells are built from biomolecules all the way up to the idea of an organism. So just to give you some context about where this fits in the year. And our essential questions in this unit have to do with metabolism. How do cells make the molecules they need and get rid of the ones that they don't need? And what happens if steps in this process aren't working? So as you'll see, this is not a traditional biomolecules unit, because it's the second question has to do, of course, with mutation. So whereas you may have taught a biomolecules unit before, you might not have taught a biomolecules unit in the same unit along with DNA and protein synthesis and mutation. So that's one of the reasons why this unit is relatively lengthy, because it takes things that traditionally are in 2 totally separate chapters, totally different sections of a textbook, and kind of puts them together. But you'll see that why we've chosen to do that. So there are 3 NGSS performance expectations that are covered in this unit, and you'll see them here. Basically, there's the one about biomolecules and metabolism, that's LS16. LS11 is the central dogma, DNA and protein synthesis. And then LS17, which is touched on in our engineering project, is the respiration standard. It's not completely covered in this unit, but it is covered in the yogurt project, which we will talk about later. So we start every unit with this is phenomenon-based teaching, right? So we start every unit with a phenomenon, which I'm going to detail for you here in our first task set. So the units and the tasks have been renumbered. So if you have been using patterns for a while, you might be familiar with we used to use U2, TS1 for Unit 2, Task Set 1. We've just simplified that to 2.1. So in 2.1, we loosely try to follow the 5 E's over the course of a unit, not in one lesson, if you're familiar with the 5E model. So in the first task, we're introducing students to the phenomenon, which has to do in this case with a young child who's born and his parents take him to the hospital within the first few days because they just have a sense that things are not quite going right. The baby Matthew scenario, actually, we adapted it from the National Center for Case Study Teaching and Science, which now used to be housed at the University of Buffalo in New York. And then NSTA bought or I don't know if they bought or were given the rights to all of those case studies. So that's why I put the link to NSTA there. Just a side plug, I would love to know, put it in the chat if you're going to OSTA this weekend in Gresham. I would love to meet you in person. Yeah, Brian, I agree. The case studies are great. We use a couple of them in bio, but I know that I have colleagues who teach IB biology who use them. Yeah, the $25 for the year actually helps you pay or just pays for the keys. Anybody can access the materials or of the actual student materials, and what you're paying for is just the teacher side materials. Although that may even be different now that NSTA owns it. You might need to be a member of NSTA, but I would really recommend that if you're not a member of NSTA. So after the students get introduced to the scenario, and I'll just show you what that looks like. So there's a story. It comes right from the case study. The idea is that we have some new parents, and they realize that something isn't quite right with their newborn baby, and they take him to the hospital, and they bring along a diaper because they notice that there's something a little bit odd about the diaper. And so they bring it to the doctor and ask, is there something, that you notice that's weird about the smell of this diaper, doctor? Because we thought it was odd and we wanted to bring it. So I have students read this scenario. We read it out loud in the class. We do popcorn reading, but there are lots of other reading techniques that you might choose to do. And then from there, I have students think in their groups about what is meant by this idea of failure to thrive, because that is actually a real medical terminology, right? And I actually get to tell the story about, I have a child who was actually almost given this diagnosis until it was determined that there was a difference between measuring in pounds and measuring in kilograms. So it's one of those stories like the NASA thing that went off because somebody measured in millimeters and somebody else measured in inches, and it went off into space because it didn't get the right trajectory. Well, my child was almost diagnosed with failure to thrive because someone wrote down kilograms with LB next to it. And so my child was considered to be severely underweight when they were not actually severely underweight. And so students get an opportunity to talk about these questions with their group. And then we talk and write down some class ideas on the right-hand side. And then I ask them to figure out some questions about what they think they would want to know if they were in this hospital. What do they need? What kind of information might be helpful for the medical staff to help figure out what is happening? And then they work with their group. After each student has a question, they work with their group to identify the two most important questions, and then they share those questions with the class. We use these prompts. Sometimes they say similar questions, and that's okay. I allow them to share the same question. And we post those questions into a Jamboard. I'm going to go back now to my slide deck for today. What I was just showing you was from the Baby Matthew Scenario 2.1 slides. And I have students build their question board, driving question board in a Jamboard because it's a permanent thing. Like we can go back and refer to those questions later on. I linked here an article from 2008 from the Science Teacher magazine about driving question boards and how helpful they can be for students to help them keep going with a phenomenon that gets presented. Let me just click on here and show you an example from my class last year, the Jamboard that my class generated. This group decided that they wanted to color code. Not only do we share the questions, but then we also group them based on the type of questions. So you can see that here, each group identified their question by their group number. And then as a class, they work together to reorganize the questions to try and keep them the same similar kinds of questions grouped together. I know some teachers who take this then and print it out and have access to it. If you do it, prefer to do it without Jamboard, if you want to do a low tech way, you can do the same thing with sticky notes. Before Jamboard existed, I used to take just a regular whiteboard and have the students put the sticky notes on the whiteboard, and then I would have one whiteboard per class, and we could go and address each of those, look at those questions or refer back to them class by class. So the idea here in this driving question board lesson, right, is or it's like about a half a period where I'm on a block schedule. So it does not even take a whole class period, but it's to get the students talking, right? And we'll talk a little bit later on about other ways that students are going to interact with each other and do some student talk during this unit. But this is a low stakes way for them to get their questions heard. Okay. I'm curious, maybe you can raise your hands. Anybody have done a driving question board before? Yeah, a few. Yeah, it's very Open SciEd has a driving question board at the beginning of every unit, and we have them in not every unit, but from physics all the way through patterns physics all the way through patterns biology. It's something that's done kind of like on an alternating basis, not every single unit, but it's sprinkled in. I like it. It's like I said, it's a low stakes way to make students sure that their voice is being heard and that their questions are being raised.
Yeah. So one thing that's sort of interesting about that unit is that, or that piece of the unit, is that there is no answer. So at that point, they kind of get left. I am terrible at it. I usually give up the answer way too early, but you don't. You just kind of go through, and the kids have unanswered questions, and then you sort of move through. And so in the course of the entire unit, they figure out what baby Matthew has. But as I said, I'm kind of terrible at it. I do a lot of.
Well, shh, don't say it now. Don't give it
Oh,
away.
I'm not saying
I'll tell
it.
you later.
But I do know what it is. And I want to tell you, but I won't. All right, 2.2. So 2.2 is about biomolecules in general. Depending on when you teach biology at your school. Some people still teach biology at the freshman level. If that is the case, then you need to do some sort of chemistry sort of, I don't know, like a review. I do like a chemistry in one day because they won't be able to handle this biomolecule section if they don't know a little bit of chemistry. But most of us probably teach chemistry before bio. So the first thing we do is we do this thing called an inquiry cube. It's something that kind of fits into all three of our units, so or all three of our courses. So physics has it, chemistry has it, and so does bio. They all have a cube. And so here's my cube. And so if you'll start it there, you can see the little video of the cube. And so this cube is sitting on the desks around my room, and I usually put a beaker over the top of them so they cannot see the bottom. And so what they're looking for is they're looking for patterns on this cube. And so they look at the structures that are on there, they look at the words that are on there, and even the numbers that are on there, and you can kind of see it floating around there. And then the bottom is blank, or it should be blank, but they can go ahead and, yeah, I totally put the beaker over the top because kids like to cheat. So I just tape it down on my desk so they don't look at it. But I also have, there's a couple of them on there. You can kind of see, Charlotte, if you'll click on the blank bottom one, animation up by Inquiry Cube. So I made these cubes during distance learning so that kids could do it over distance learning. Obviously, I wasn't going to mail them a cube. But if you go to this website, you can see, and you can actually grab it, Charlotte, and move it around. And so the cube is there, and I use this actually for demonstration just to kind of show them. And then there's a completed cube. That's the other one that I use to sort of show at the end what is actually on the bottom. So on each of these, they have a name of some sort of monomer, right? So the piece that makes up the polymer, so the bigger piece. So you can see amino acids on one side, and on the other side is protein. On one side, it's carbohydrate. On the other side, it's sugar. And then on the top, it says nucleotide. And then on the bottom, they have to figure out what it says. And so you can see that D there on the bottom right. That is the first letter of the thing on the other side. 2 represents how many words, and 3 represents how many letters. And so the thing on the bottom has 3 letters. It's going to be yellow. It's going to be a bigger piece or a polymer of a nucleotide, and it's going to start with a D. And so, like, you know the answer, I think, but
Yeah, the two on the bottom left is the hardest one. It's actually the number of letters that the 2 words share in common.
Oh,
So for
really?
example, yep,
I always
protein.
thought it was how many words they were together, because that works too.
Oh, okay. Well,
So
that works
amino
too.
acid and protein is
So of
3
course,
words
here's the
total.
shocker. Here's the bottom, right?
Yep.
But I actually have a little piece of paper stuck over over mine that say, "You weren't supposed to look. You were told not to look."
Mm-hmm.
And then, the longer they read, the more likely it is that I'm going to see them looking. But I love the idea of the beaker. So
Yeah, the beaker and the tape, because you can hear the tape from a mile away. And I'm like, what are you doing? But no. And so that's kind of the start. I mean, it is patterns biology, so it makes sense to kind of start with some patterns in this particular unit. Other things that we have on here are just some pattern matching. And so there's a bunch of different ways to do this. You can use paper. It's like a card sort. And so I give them envelopes with the different molecules on them, and they try to separate them out into carbohydrates, proteins, lipids, and nucleic acids. And I think there's even like a energy producing molecules piece. And then there's also a teacher Desmos version.
oh, let me show you that.
Yeah. So this is something I put together again during distance learning because I couldn't pass out paper. And so this is the more advanced version of it, but they put them into groupings. And if you do control minus, Charlotte, it'll make it so that it's a little bit better. Yeah.
I got it now, I think.
Yeah.
So they can group them into categories. And Jason's great with the Desmos Classroom or Teacher Desmos things, and he's added in all the terminology, right? So these molecules are the ones that are the originals to this task, but he's added in, pictures of food and names of different molecules and things like that. So you can, yes, this link is on the outline, Karen. I don't think so, because we just added it to this slideshow. So we'll make sure that we get it on there. Yeah, I can do that while Jason's talking.
Yeah, so I actually have that version, and then I also have a more advanced version that I use in AP because we do a similar thing in AP. But we do like to use the paper whenever possible. I like to get them off the computer whenever possible, but sometimes it's just nice to use that as sort of a warm up or an activity to just get them sort of thinking. Yeah, I think that's it for that one.
All right. So as they continue on with biomolecules, right, it could very well be that this is the first time that they've actually heard the word carbohydrate without specifically having to do with food, right? So I like the fact that you can make this connection, and I'll tell you about a possible assessment related to nutrition a little bit later that makes that connection a little bit more obviously. So once they have the basic ideas about the different four different categories of biomolecules and the characteristics that they have in common. One thing that I did want to actually point out, I wanted to show the example of the Jamboard version of this. Since we talked about Jamboard earlier, let me just go to another panel here. Students can actually pick up the pieces and move them around on the Jamboard. And then my students have a touchscreen Chromebook with a stylus. And so I asked them to circle the similarities that the molecules have, right? So here you can see they've labeled this amino acids. This is kind of after we've discussed at the end. But before that, one of the things I asked them to do is let me go back here to the instructions, to pay attention to what elements are in the molecule, what the shape is, patterns they see, and what the ends look like. And when they do those things, they are actually able to notice that here, every single one of these has a COO minus, right? And I call that carboxyl. We talk about that, but that's not something I'm going to test them on. But it's a way for them to be able to put these molecules together. And then we eventually talk about that as an acid group, as opposed to the amine group on the other side, and we get the word amino acids, or that term. And then so on. There's like all these different kinds of things, all these different annotations that they can make on the Jamboard because they can use their stylus to write. And so they've used, in this case, this group identified the characteristics that were in common using a little sticky note. They circled those in their picture. They wrote some things down, and then eventually they identified like that this group down here were also carbohydrates, but a different kind. They're fats as opposed to simple sugars.
Yeah, with the paper version, a lot of times I'll use those chalk markers and they'll circle them and they'll label things that way. And they love that. Some of them draw little pictures and things also, just because it's neat to write on the desk.
Yeah. So there's a lot of different ways that you can do this task. This is from an old AP Bio activity that's been on the web forever, but we've not ever been able to find who originated it. So if you know, please let us know. We would love to credit that person with the original work. All right. So once they have some basic terminology, we start to talk a little bit more in detail about the metabolism, because we know as teachers, we know that the metabolism, the making and breaking of biomolecules, is the most important component of what we're trying to get at, because all those molecules have to work in consort in order for a cell to function. So the next task is about the functions of biomolecules and how they actually get to be put together and broken apart. And there are a couple of different ways that you can do this. Prior to COVID, I always had students do a building and breaking of biomolecules activity with physical models. So building a glucose molecule, for example, out of a modeling kit, and then attaching two glucoses together and recognizing dehydration synthesis, and then breaking those two glucoses apart and doing hydrolysis. And then contrasting what a glucose molecule looks like compared to a very basic amino acid, right? The different kinds of atoms that are in there. It's reinforcing some of the ideas that they saw in the card sort. But post-COVID, well, during COVID, we couldn't do that at all, right? Building of kit, using a kit from home, that wasn't possible. So I decided at that time to dial it back a little bit and really I felt that sometimes my students were getting lost in, what is that expression? Getting lost in the forest despite the trees, right? They were focusing, they really enjoyed building, but they were getting lost in the details and not really getting the ideas of building molecules together to make polymers and then breaking them apart, digesting them to make the monomers when that's necessary. So I shifted during COVID to a less detailed, more generalized video and assignment. Thanks to Rachel Taylor, who is a teacher at Millennium High School in New York City. She posted her tasks for this topic in the NGSS Biology Facebook group. And we borrowed those activities and have put them together. And I linked just her, one of her videos here, which I'll just play it here, but the student work actually follows directly, but I'll just play a little bit of her clip.
When atoms join together, they form molecules. When carbon, hydrogen, and oxygen atoms join together, they can form organic molecules. Organic molecules are the molecules found in living things. 4 major types of organic biomolecules include carbohydrates, DNA, fats, and proteins. These molecules contain the elements carbon, hydrogen, and oxygen. Some contain nitrogen and phosphorus as well. Let's take a look at the importance of each biomolecule in living things.
So I'm going to pause it there. But what's amazing about this is that she did all of these videos. She did these videos, and she has a channel, but all of her student documents have the same images that are in the videos. So she's clearly done all of the images herself and built her student work in the same turn, or at the same time. So I really liked that. That worked very well during COVID. And then I've continued to use it since we've been back in the classroom. And I find that the students have a much better idea of those terms, synthesis and digestion, and that they realize that we're not just talking about digestion that happens in our digestive system as human beings or as mammals, that we're talking about the building up or breaking down of any kind of molecule in any organism. So it is less detailed, but I have found that it's been very successful. So that's an alternative for you. Jason posted a link to an online ball and stick model website where you can use as a demonstration, or if there are students who are absent who miss the ball and stick models in class, they can use those. So this is about a third of the way through the unit. There are 9 tasks, and this is the third one. I like to do a check-in with students at this point. There's a quiz here. I think that in the unit outline, we suggest that this quiz be a formative grade, that it not be something that count towards their grade permanently. You could break this down into individual questions. Like if you have a student on an IEP who gets an oral test, you wouldn't have to give them every question here. Or you could use those as a opportunity for retesting orally. I give this quiz, but I have students just use it as a check-in. We correct it together. I don't often do that, so it's kind of a fun thing for them to be able to do, since that's not my usual way of operating. But if you wanted to do a more nutrition-based assessment, we developed, my colleagues and I who were working together during that CDL year, we developed an infographic assignment where students were taking this information about biomolecules and applying it to nutrition. And then they were building an infographic. So that's another opportunity for you if you would like to assess at this point and have it be a little bit more real world. It's not as connected to baby Matthew per se, but it gives you another opportunity to assess at this stage of the game. Questions? Now we're getting into the real meat. So lots going on in the next task set.
Yeah, so this is, as Charlotte just mentioned, probably one of the bigger pieces of the Unit 4 or Unit 2. So it's Unit 2.4. How do organisms make and break molecules? It's sort of this idea that I think we always mention you are what you eat. And you can see that there at the bottom. Like, what does that mean? And so you are physically made of these things. So does that mean, and you can see over there on the right, that moth, since it eats a lot of sugar or whatever, is it going to taste sugary? And that's just not sort of the case, right? So how do these organisms make and break these molecules? And the answer is
Yeah, my
enzymes.
students always get a kick. These in the slides, these pictures actually show up one after the other. And they always really respond when I say, but if you ate a moth, and then they're all like, ooh, gross. So
We've all done it.
This is an attention getter for sure.
Yeah, so if a moth only eats sugar, but the moth is not made of just sugar, would it not taste sweet? And they were like, yeah, it kind of throws them for a loop. So there's a couple of hands-on activities in this one. One of them is pop bead ace or toothpick ace. It kind of depends on how you want to do it. I do the pop beads. They are fairly economical. If you'll click on those real quick, Charlotte. They're not bad. So they're basically, I think young people use them to make necklaces. They're these little beads that sort of pop together and you can make these fancy little bracelets and necklaces. And I think you can buy them for like $20 for hundreds of them. Like they're very inexpensive. And so what they're doing is they are measuring the time it takes to pop them apart. And so there's a bunch of different intervals that they do, and then they try to do it with like one hand versus the other. And then at the very end, it even mentions what would happen if you were to put your hand into ice and try to break these, or if you put your hand into like a big old oven mitt and tried to break these. And so we talk about some of the things that make enzymes not function correctly, right? Denaturization and so on and so forth. But it's super fun. They get really competitive with this. They want to pop as many as they possibly can. And so that's what this is. So again, this is a version of the toothpick ace. The problem with the toothpick ace that I don't like is that it's just breaking toothpicks and then you throw away a bunch of toothpicks. And this one here, you put in a bunch of like paper clips and they kind of get in the way. And so the students have a difficult time finding the pop beads. But what happens is as you pop them, once you run out of substrate, you no longer have them. And so obviously you slow down. So the graph goes horizontal. So it usually starts out very linear and it's going straight up. And then all of a sudden it goes horizontal as they run out of substrate, which is exactly what happens in a regular enzymatic reaction. And so you can kind of see a substrate up there. You see sucrose, which is glucose and fructose together. Kind of looks like two pop beads next to each other, right? So they're popping those two things apart.
I'm just looking for my Desmos graph that actually shows the results. So here's what the results look like in Desmos for part A, right? So this is a good opportunity for them to practice with Desmos with one of the two-phase graphs that they will get actually when they do their enzyme lab. So it's a good opportunity for additional Desmos practice.
Yeah, and as I mentioned, like there's a lot of them to pop at the beginning, so the rate is very quick, and then all of a sudden they have nothing left and it flattens out and goes horizontal. Do you do toothpicks or do you do pop beads, Charlotte?
I use pop beads. I don't necessarily do it every year. Last year I had to be out of the classroom right around this time for a family emergency, and so we did it. We'll have to see. I need to catch a couple of extra days to be able to finish Unit 3 by the end of first semester. So we'll have to see. I'll have to talk with my colleague to see if it's going to work for us this year. Plain beads or mixed ones? What do you mean, Leah?
Mixed in color?
Like some are different shapes and stuff. Do you just buy the plain
Yeah, I
common
do the ones that are
size?
all the same shape.
Yeah, they're all spheres with the little poker out the end.
Gotcha. Thank you.
Some they need like 100 beads per pair of people, per pair of students. So like the huge ones that you might have had when your kid was a toddler, right? That's not economical. Though I bought when Mountainside opened and we're buying all of our materials. I think we bought like 2,000 pop beads. So, and I quickly realized that I just needed to count out 100 once, and then I had a TA measure them out by weight after that.
Yep.
So another thing that I like to do during this that helps to solidify the connection to baby Matthew later on, this is kind of like I do this now so that they have a better chance of being able to effectively explain what's going on with baby Matthew later, is that students do research on some metabolic enzymes. So we've obviously defined what an enzyme is, but they don't really understand what enzymes do until we take a look at some of these examples. And so I let them select, let me just get back to the example. I let each pair of students select an enzyme from this list. You can see that this is an example that they've already written their names on from last year, but they get to select one, and then they have some instructions and a word bank. And then I give them an example, which happens to be with catalase, which is the enzyme that we investigate in the lab that's upcoming. They don't know that yet. And then I provide them also with a template that, shows like what kinds of things they're supposed to have on their slide. And I've done this as a filler, but I've also done this with a substitute. This works out well. And then each pair of students has something that they're specifically investigating. They can't copy from each other because they're all doing something different. And we can come back to this later to refer to the different kinds of enzymes that are responsible for different parts of the metabolism. That is like a research-based thing. And then there is we've written just a quick check-in quiz. This one is listed in GoFormative, which is now just called formative.com, enzyme structure and function. I use a quick Canvas quiz as a check-in, just so that students have an idea about where they are in terms of the content. I don't score it for a grade, but I give it to them as a formative opportunity to see where they are.
And then we do one of the bigger labs that we have for the whole year, which is the enzyme lab. And so this is taking potato ace, which is like a catalase, and you are basically breaking it down with hydrogen peroxide. And so what happens is they put the two together and you split them into groups. So the groups are going to be temperature, right? So there is certain groups that are measuring the same substrate and the same enzyme together in a certain different temperatures. I think there's a cold, there's like a room temperature, there is a hot, and then there's like a really hot. Yeah, sorry, I said it backwards. Hydrogen peroxide is your substrate you breaking down. I just did a lab that was the opposite of that, so it totally messed with my brain. And then you have the other one is pH. pH is actually the hardest of them to do. So you have to measure the pH by adding, what is it? Sodium, no, yeah, sodium hydroxide and hydrochloric acid. So you're adding those to it. The hard part is sort of measuring the pH. So you can either use pH paper or you can use vernier probes. That's what we do, but it's kind of inconsistent. So I know a lot of people take that one out or give that to them as an example. Then the other one is substrate concentration. So you dilute down the hydrogen peroxide. And then there's the enzyme concentration where you break down the potato catalase with distilled water. And so the instructions are all there. It's one of those labs though, you really have to be super organized. You want to set it up around the room and all the groups are going at the exact same time. And it is a perfect opportunity for them to do a data discussion to explain what they did in their particular group. So we go around and we do little presentations because everybody can't do every single experiment. So they only do the experiment that they've been assigned. And so usually in my classes, I have about two groups doing each thing. So it kind of depends on how many kids you have in your class, but it's usually groups of 3 or 4. And we have, as I said, 2 groups per. And there's actually a sort of a demonstration that I do to begin with, because what they're doing is they're measuring, and you can see it in the picture here, they're measuring bubbles. And so one of the things I do first is I do surface area. So if you take that potato and you cut it into pieces, and so in an A, for example, I would put one single potato piece that's the same amount as in all of them, but the next one is cut in half, and then it is cut into fourths, then it's cut into eighths and so sixteenths. And so what it does is it gives you that same sort of graph, and it shows you that the surface area actually has an impact. And so I show them how to measure the bubbles and all of that stuff. I do a lot of those little sort of demonstrations before they go in, just so they know what they're looking for. So in this particular lab, they're looking for how high the bubbles get, because that's the oxygen being released. And so they want to have some sort of example of that.
Yeah, so one of the things that Jason touched on this a bit ago, but if you've taught patterns physics or you taught patterns chemistry, you're familiar with this idea of having really truly multiple labs happening in your room at the same time. And that is the crux of the patterns approach to inquiry, is that students are not all doing the same thing, and that there's not just one answer coming out of the lab. And not only that, but they are responsible for sharing their information in the data discussion such that the other pieces, the other groups, everyone shares, and but at the end, everyone knows how all of the different variables affect the outcome. And so the idea of a data discussion, I'm going to put in another shameless plug for OSTA on Saturday. Christy Miller and Catherine Kraft are going to both, who are chemistry and physics teachers respectively, are going to do a talk on Saturday about how different types of data discussion work in different types of lab situations. So in this case, I often do a jigsawed data discussion where I don't actually have to do much talking at all. I group the groups, or group the kids together in groups, so that they always have a partner with whom to present, but they're presenting to a smaller group of students, and then they get their questions answered about their different, or they get questions asked to them about their results, and they have to communicate back. So it's another opportunity for students to communicate to each other in the classroom, really prioritizing that student talk that we all want to happen.
And 2 of the groups share the same graph. So substrate concentration and enzyme concentration, they both get a linear graph. pH and temperature get what's called a Gaussian curve, which is sort of like a Goldilocks thing, where it's in the middle is great, but both of the outsides are terrible. So like too hot, too cold, just right is in the middle. So that's what Gaussian looks like. It's like a little mountain. But yeah, the kids all get it, and it's one of those days. Like, it takes a lot of prep, to to teach science right takes a lot of work, but it's very rewarding because the kids are getting really, complex concepts, which is kind of why I like doing some of these things.
We do have some videos of these experiments. Those some of you who know Jessie Chamberlain will recognize her in those videos. We have 2 trials of each of the 4 variables so that you could have students even take an average between trial run 1 and trial run 2. They can see what goes into every tube, and then we have not measured for them. We actually did these videos during CDL and created the videos and edited them such that we held up a ruler right to the tube so that the student can pause the video and actually do the measuring themselves, right? Rather than just being given the value of how the height of the bubble column in the tube. So if you have students who miss, those resources are there.
Yeah, we can add this also later. This is another one of those teacher desmoses. This one is set up so that it graphs it for them. So again, if you have kids that are absent, this is Jessie Chamberlain doing her video, and then the kids follow along and write down the numbers. It graphs it, and then they end up with the same results as the kids that were there.
Any clarifying questions about Enzyme Lab? I don't have your faces in front of me, so hoping that nobody's raising their hand. There we go. I got everybody now. Okay. Oh, so the way that I like to close on enzymes is to, close. I say close on enzymes, close this task set really, because we, of course, end up talking about enzymes for the entire rest of the year, is to give students a connection between enzymes and the metabolism on a bigger scale. And so Jessie Chamberlain and I, during COVID, we put together this Jamboard where we show students the pathway that a cell can take to actually convert a glucose molecule into a glycine. And we researched this pathway. It's a 9-step pathway. And we identified as I know it's a little hard to see there with the screenshot, but you have the glucose molecule, and then the arrow, of course, indicates that glucokinase is the enzyme that generates the next molecule, which is named as glucose 6-phosphate. And on the next panel of the Jamboard, and you can look at this, if you click on the link, it will show you the template that I give to students. They basically have to look at those other molecules and figure out which one fits in each circle. So they have to identify what the product would be. And then that also helps your high flyers. We often have high flyers who kind of can figure out puzzles like this relatively quickly, but it gives them the additional piece of information about the naming of the enzymes. So if they look closely, they will see that kinases are always enzymes that add a phosphate group, and that a phosphatase would be an enzyme that would remove that phosphate. They can see when a hydrogenase or a dehydrogenase or a transaminase, what that would do, moving an amino group from one side to the other. So let me show you what this would look like when the students have finished with it, because it looks similar to what we did with the other one earlier on. And I like Jamboard also because I can go in and write notes on their Jamboard. So I tend to bring my iPad that day, and I use my iPad with my pen or my pencil to write notes to them. So I highlight and draw arrows, and I wrote this note. Careful, this was a pentagon. I think they ended up changing it. So obviously they have a hexagon here now. But I asked them to circle the parts of the molecules that got added or removed. So in this case, the phosphate group was the thing that was added onto a glucose in order to get glucose 6-phosphate. And then here they can see that you go from glucose 6-phosphate to fructose 6-phosphate, and the phosphate's the same, but the sugar is now 5-sided instead of 6-sided. So this is doesn't take very long, but it allows students to see that there is a pathway of enzymatic reactions that happen in order for a molecule to be changed from one thing into another. It's not just a one and done. And I have students like working on this simultaneously. So every group has a different panel of the Jamboard that they're looking on. All those panels are the same, but they're working with their group to kind of figure out the puzzle. Okay. All right, so we do two pretty big lab activities, basically back to back. And we used to put this is the yogurt engineering activity at the end of the unit, but it felt really disconnected. So about 2 years ago, the first year that we were back in person, fully back in person, was that just last year? I don't know. I decided that I wanted to try this in the middle of the unit. Now, admittedly, making yogurt in the middle of the unit and transferring to or switching gears to respiration from enzymes is a little bit of a disconnect from Baby Matthew scenario, but it does the best job, I think, at representing what a enzymatic pathway does. And it's interesting for students because we produce food, which so many of our students have no concept of where their food is actually coming from.
Yeah, during the summer, we did both of these on the same day. I don't suggest doing that. Enzyme lab and yogurt lab should be done on different days.
Yes. Note to ourselves for every future summer workshop, we should never do these on the same day. Too much. It is also, it's a high prep experiment to do, but I run it as an engineering project. So engineering, of course, as all of you know, is one of the required things of the NGSS, and it has its own standards. This project does not hit all of those engineering standards, but it does allow the students to design a solution and actually to create the solution and test the solution. You could do a second round if you wanted to, but that would add another several days to your process. This actually originated with me making yogurt at home. We eat a lot of yogurt in my family, and it was getting kind of pricey. And I think I saw a YouTube video that told me that I could do it on my own. I tried it at home and then ended up developing it with a colleague into this student project. So the students take on different roles in this project. You can see them there: food scientist, quality control engineer, testing specialist. And they get to decide in the scenario, it's the cafeteria, their school cafeteria, that's looking for a new yogurt vendor, and they're looking to improve the composition of their yogurt and keep it under a certain budget. And so the students get to decide from a list of ingredients, and they learn what the different characteristics are of the different kinds of ingredients, what fat does to yogurt, what protein does to yogurt, what length of incubation time does to yogurt. And then they make some decisions in their group about which ingredients they want to use and how long they want their yogurt to ferment. And then the way that we tie in respiration is that we use the respiration pathway of the bacteria, which is, of course, the fermentation pathway. So that's how we bring in the respiration standard, is by having them write about fermentation. And then, of course, they do the fermentation or allow the bacteria to do the fermentation. In the past, if you have the opportunity, I would love to see someone take this on or do it. If you teach engineering more deeply, or maybe you have some students who are taking engineering alongside their biology class, especially some of you who are in small schools where sometimes the kids kind of move together from class to class, I I have an extension that's available for kids to be able to build and test their own incubators. I worked with Mahir Ravel to develop, they figured out how to, or he helped my students he came in and helped my students build build a heater that had an on and off switch. And then they also built a housing for that incubator so that the heater didn't have to be on all night long. It actually, they tested the different insulating materials and the success of their incubator based on how much electricity it ended up having to use to keep the yogurt at the fermentation temperature all night long. So if you're interested in that, I can get you the materials, and I even have some physical materials that I could get to you for building of those incubators. So there are several different ways that you can assess this task. You can assess it on the different components of the engineering design cycle, the engineering standards. You could assess, there's kind of a mini lab built in. The students are along the way collecting data, but you could have them write a lab conclusion on the question, what is the effect of incubation time on the pH of yogurt? They collect data as a class, and then that they can graph those data and then come to a conclusion on that. The students do testing of the yogurt. They test the viscosity. So that brings in some ideas about force coming back from physics. Obviously they test pH, and then we do give them an opportunity to do a taste test and to rate rate their own yogurt and that of other students. That's always a fun day. I certainly don't force anyone to do a taste test if they don't want to, but every year I have some students who come back at lunchtime and say, can I take that at lunch? What's left from my group? What's left from my class? And I've had students who I've run into after the fact, 2, 3, 5 years down the road. And this is, for me, one of the things that they really remember doing. During CDL, we had an opportunity, with some colleagues in North Bend who had written a similar project about sourdough bread. And so we had some students who chose to do bread instead of yogurt during CDL, but I did have some students actually go through the process and make the yogurt at home during that time. So it is a memorable experience for them.
I think in my experience, I find teachers are scared of this one more than anything else, but it's also probably the most impactful. Like I have kids come back and like, what was the recipe again for yogurt? I'm like, well, let me look up what you did, and I have all those, but it's crazy. They really enjoy this one. This is the one they remember the most.
I'm
But
curious,
it's
some of
tough.
you have, yes, I'm curious, some of you who have done this in the summer class with us over the years, have any of you tried to do this in your classroom? I'm wondering, like Katie or Kim, Monica, do you guys do it at Southridge? You can put this in the chat. I was just curious. Okay. Oh, great. Cool. And Kim, do you find that it's a good one? Do you find that they like it?
I have kind of mixed reviews about the yogurt lab, but they definitely remember it.
Mhm.
Some of them are a little afraid to try it, and others are like excited, but yeah, they definitely remember it.
Yeah, we have, over the years, I've built pretty detailed teacher notes. Thanks, Kim. Pretty detailed teacher notes about this project. For me, the number one thing that kind of makes or breaks the results of the taste test is the starter culture that you use. And I have found that the yogurt that I might like at home is definitely not the yogurt that I should use as a starter culture for students. Because I eat Nancy's yogurt at home, and that is much, much, much too acidic for students, even if they put in the maximum amount of sweetened condensed milk. So I tend to stick with a very neutral Greek yogurt when I'm doing this with students, and that produces generally some good results. Oh yeah, Karen, that would totally work. I use a sous vide machine, but but you have a much smaller number of students. So 9-cup yogurt makers, that gives you access potentially for 18 groups. Some of your classes are tiny. Each kid or each pair of kids could have their own sample.
And our kids are so used to eating really sugary foods that on that day I do bring in granola and I bring in like jam, and that really spices it up. Obviously, they have to do the regular taste test first, but
I have 2 classes in between them. It's a total of 18 kids. So, yeah.
Yeah. That'll be great. And
yeah, what was the
as long as you're willing to take them out at the right time or set them in so that they'll all be finished at the same time.
Kim's comment reminds me, Charlotte, that you said that there's one yogurt that is terrible to use as a starter.
That exactly what Kim just said.
It's Nancy's.
Yeah, it's what I eat at home and I don't mind it, but it's awful to use with students because it's very, very acidic.
Yeah.
Yeah. So Greek yogurt's much, much better. We get much better results. Any other closing comments or questions about yogurt? It is really fun. And if you have any questions about how to do that, I've done it every which way, and I'm happy to answer any clarifying questions that you might have. If you're considering it, I would love to answer any questions that you might have.
All right. So speaking of food, 2.6 and 2.7, protein synthesis, we use sort of the analogy of a restaurant. And so if you'll click on that real quick, Charlotte, the protein synthesis down there towards the bottom. So we start off, and I always ask the kids a question. I like, pretend like I've never been to a restaurant before. Sort of explain to me this process. And so the kids all, have been to a restaurant. They tell me sort of, you go in, you sit down at a table. And then I said, well, what do they do? Do they give you anything? And they tell me that they give them a menu of sorts. And then I ask them, what is that menu? That menu happens to be the DNA inside of the cell. And that is what is going to be leading to the building of whatever you have ordered. So if I'm ordering a cheeseburger, that cheeseburger is going to be put together in the kitchen by, in this case, the tRNA, right? So you have your DNA, which is going to be taken the order. So who comes to your table? Some sort of messenger / waiter, waitress, and they take your order. And do they make your food at your table? Unless it's Benihana, probably not, right? They go to the kitchen, which is the ribosome. In the ribosome, there is the tRNA, which is going to put the individual ingredients, otherwise known as amino acids, together to create your cheeseburger. And does it matter the order on which they put things together? Absolutely, right? Maybe not as much in a cheeseburger, but definitely in like cookies, right? If I bake my cookies before I put the flour in, or if I change the flour to hot sauce or whatever, it has a huge impact on the overall product that's being made at the end, the protein that's being made. And so this whole structure, so I kind of use that analogy and I talk about where is the table? They're like, well, it's out in the dining room. I'm like, well, your dining room is your nucleus. I said, you don't go into the kitchen and prepare your food. You are in the nucleus as the DNA. So something comes to you, that's the mRNA. It takes down and writes down your message. And you can see right there, it says use base pairing rules to transcribe the DNA into mRNA. So I always talk about that they write it down on a little piece of paper, but they write it down in their own little sort of language. So it is an RNA language. And so the next 3 there would be GGU. And then they take that and they take it to the kitchen. And then the tRNA is going to come back with, what did you say? You wanted GGU? And they write it down as CCA. And then that CCA translates into a particular amino acid. And so we kind of go through that as I wouldn't call it a lecture, but it's definitely sort of a back and forth where I'm talking to my kids about what is occurring. And then they go and they do activities like this. If you go down a little bit further, you can see that they can use the little circle thing there to basically translate into the particular amino acids based on the letters. So from the middle out. So AGG would be arginine, for example. And so they use this as sort of a, you know, it's a worksheet, but at the same time, they're kind of understanding how to put this together, and using that analogy of a restaurant is something that they have some scaffolding about, and they understand how that functions. And then before that, you talk about DNA just in general. You talk about Chargaff's rule, which is A's go to T's. He found that by looking at a bunch of different species and noting that their A values and their T values were almost identical in percentage. And then DNA structure and protein synthesis is just another alternative. And then we also go into mutations here. So there are some mutation things that we look at. So we look at them.
Hold on just 1 second. I think that's 2.8, but
Is that 2.8?
yeah, but I wanted to actually point out about the less detailed alternative. Because Jason, you mentioned tRNA in there, and all of us as biology teachers, we know that that's the case, right? We know that that's what happens inside the cell. But believe it or not, the NGSS standard for this topic does not call out all of those details of A site and P site and tRNA versus mRNA and how the ribosome fits together and Tata boxes and all of that. Right. So I actually have decided to kind of pull back on the level of detail that I present to students. And so I like to use this less detailed alternative and skip over the idea of tRNA, especially because the decoders, like the circle decoder that we were just looking at, or the more traditional rectangular decoder, they actually don't even use tRNA. Those are all read using mRNA codons. So we at regular biology level don't really talk about tRNA except to maybe say that it exists, or to answer a student question if they've read or see something on a website or a book where they ask about it. Obviously, if they're taking IB or AP biology, or if you're using patterns coursework as the basis for one of those upper level classes, then you don't have the luxury of making that decision. But just know that that level of detail is not covered in the NGSS standard, and it's not in the performance expectation that students should be able to detail all of those specific inner workings of what's happening in transcription and translation. So I can just pull that up. That one's clearly a wrong link, so I need to fix that. Maybe Jason, and maybe you can add that to the parking lot, that I need to fix that combo doc or a note.
Will do.
All right. So especially with pulling back on all of the details of the central dogma, really what I think is one of the things I think is most important for students in a biology class is to understand how much Hollywood really is trying to mislead them when Hollywood presents things about science. Especially this word mutation. Hollywood leads kids to believe that one mutation is going to cause them to, I don't know, sprout a third head. And as biologists, I think it's one of the biggest responsibilities we have is to help students understand how some of the words that are thrown around in everyday language are actually misrepresented in terms of science. That those scientific words are used, but they're misrepresented out in the real world. So I actually like to spend more time talking about the ins and outs about how mutations work, rather than the ins and outs of how the protein is made per se. So like we talked earlier about pulling back on some of that detail in the previous lesson, but here is where they actually need to know the impacts of when things don't go correctly. So they first investigate the effects of mutations in the context of the condition muscular dystrophy. And so we focus on the protein dystrophin. Oops. Control Z. Let me pull this so you can see that properly. So this protein dystrophin, which is represented in the upper right-hand corner over here, is a connector between cell membrane proteins and structural proteins on the inside of a muscle cell. And when dystrophin doesn't have the right shape, then those two things are not connected properly. And as a result, the muscle does not contract. The muscle cells don't work together to contract the muscle effectively. And so the first thing that the students are doing is practicing again the same skill of transcribing and translating that they did in the previous lesson. So if they didn't quite get it the first time round, they have another chance to show it here. And then they can see that in this example, there are two different kinds of mutations. We have a substitution mutation, and I've identified it here. You might choose to not identify it for the students and let them figure out where the mistake has occurred and what kind of mistake it is. And then the second one has a deletion. And then I've already set up the frame shift in this sequence. So all of the blue letters are the ones that have already been shifted one position to the left. So, and there are slides that go along with this that show like a sentence in English. I think the sentence is, the fat cat ate the rat. And then you can demonstrate how, or the slides actually demonstrate, have an animation that shows how adding a letter can make a minor modification to the term, and then, but how removing a letter might have a huge effect if you have a frame shift one way or the other. And so the students, after they kind of figure this out, and I think that this second mutation has, I think it may generate a stop codon. I can't remember if it's a stop codon or if it just totally changes the sequence. But then they write a little bit of an explanation. Why is deletion worse than substitution? Right? And so they have to use what they saw in the simulation with English words and put that together with what they observe here in the example of DNA to protein. And by the way, these changes, these are actual changes that actually happen in the dystrophin gene. So depending on what kind of mutation you have or a person has, they might have a different version of muscular dystrophy. There are actually lots of different kinds of muscular dystrophy. And we're just looking at two of them here. Now, by the way, this came from an original activity that was written at the University of Pennsylvania. It's the original activity is not new, but we've made some modifications and adaptations to it over the years. So once they figure out, based on the data, that deletion is worse than a substitution, then we bring in like real world scenario. And this is, again, the connection back to, even though it's not the same condition, connection back to medicine, to the idea of baby Matthew, and how baby Matthew's scenario might actually be connected to something that might have changed in his DNA. So we have descriptions of different kinds of muscular dystrophy, and then students write a short CER where after they investigate the different kinds of muscular dystrophy by reading and watching some patient stories, they write a little CER, short one, about which mutation from page 1 is more likely to cause the more severe form of the disease. So you can assess that just for the CER writing, right? Can they take evidence and use it to support a claim and provide some science behind that as reasoning? Or you could assess this as a formative for them to say, yes, you have this, you have mastered the skill of transcribing, or you have mastered the skill of translating a DNA or an mRNA. And then I follow that up with another Jamboard. I told you early on that there were a lot of Jamboards in this unit. But I like to have students share or to share with students examples of all different kinds of proteins and what happens when those proteins don't function correctly. So there are several panels in this Jamboard, and you'll notice that each one of these blue or yellow stickies is a different color. And these are all different kinds of proteins, transport proteins, defense proteins, structural proteins, and so on. There's 8 different panels. We picked 40 different enzymes so that the largest of classes could still do this activity and not have any duplicates. And then what the students do is that each one of them selects one enzyme from the panels. And there's an example here that's the same example on each panel that shows what I would expect them to do. They search for the name of the protein. In this case, lactase is an enzyme. And then they find out, well, what happens when this protein either doesn't function or doesn't have the right amount in order to do its job effectively. And so they write a little bit of explanation about that particular protein that they have selected. And this, again, allows them to make a connection, or potentially future make a connection, that the condition with baby Matthew, or the situation that's having with baby Matthew, may be related to some kind of protein that he either has not enough of, or that he might have too much of something else, or that his body is not making at all a protein that it really needs. Now there's an opportunity for an assessment here also, traditional assessment. These things are a little bit hard to assess in alternative ways. I did have my colleague last year decided that they wanted to do a one-pager. That was another kind of assessment. If you're familiar with that kind of thing, it allows students to show their knowledge in a different way than on an on-demand exam. And so, sorry, any questions about any of those 2.7, 2.6, I guess, through 2.8, which is almost, in traditional teaching might be considered a whole unit in and of itself, from the structure of DNA all the way through to mutations. For assessment purposes, I would encourage you to break it up. Break up the unit and not just have one single test. Okay. All right. So that all of this brings us to the last task of the unit, which comes back and over the course of the unit, I should say that I check in with students at the end of every lesson about how what we have learned could lead us closer to baby Matthew's explanation. Right? So they've tracked that in their INB. At the end of every lesson, we do a reflection, and that reflection, I ask them to connect back to identify what is it that they had learned here, which can help us to understand what's going on with baby Matthew. So by this point, we have a pretty good idea that baby Matthew's condition is being caused by the fact that he must have inherited a mutation, or his cells must have a mutation in them, which is causing whatever is going on with him, right? That he has some a defect in a protein that is supposed to do something with his metabolism that is not working correctly. We know it's his metabolism because something in his excretions has a very odd odor, and the and neither of his parents are affected by this. Now, side note, we have not done genetics yet, right? So this might not be a time period where you would want to go into the, Punnett squares or a pedigree or anything like that. That comes down the line in Unit 4, but we haven't addressed that yet in this course. But it's a good setup, right? I come back to baby Matthew and use baby Matthew as an example later on. So this is one of the things that actually has a new component this year. If you were at this webinar last year, I hope you're not bored, because we've not a ton has changed in this unit since last year. But one thing that is new this year is that the setup for this task, we set it up based on the suggestions from summer class. So Elena and Monica, this came up when you were in the summer class 2 years ago. My colleague drew up an NCAA style bracket to help us with this task. And that worked last year, worked really well. So let me tell you about it. So one of the pictures that I have up in my classroom is a huge poster of the human metabolism. And as you can imagine, it has, maybe Jason, you can look it up and put it in the chat so people can see. But it's all of the human metabolic pathways. And I think it's called metabolic pathways, Jason, Good, Karen. There is a link to the poster in the unit outline. And you can buy it, but you may want to check in with, I don't know if Casey Boothby covers your area or not, but he, through the RAP grant, may be able to get you printouts. We have a poster printer, and I know like in Hillsborough, they have a print shop. You might not have that in Elkton. But I would check in with Casey, because he may be able to get you some of those things that you might need through the RAP grant through EOU. So there's this big poster, and Jason just posted it. You can kind of see a little screenshot of it here, but he just posted it for you there in the chat, so you can open it up in your on your own screen. And then there's a version of that poster that actually demonstrates many, not all, different. Oh, that link doesn't work. I'm going to have to figure that out. Different mutations that cause metabolic challenges. And we have identified 16 of these as possible scenarios for baby Matthew's condition. So let me show you the slides that we use for student support here, what students will be looking at. So our question that we're trying to answer in this section is, what is baby Matthew's diagnosis, and how can it account for the symptoms that he is experiencing? And so we review over the idea of what metabolism is and come back to the scenario of baby Matthew that we saw in 2.1 very early on. I actually bring out their Jamboard, their questions at this point, their driving question board, and we look through which of those that we have actually already been able to answer. And then we look at this. Oh, here's that poster that Karen, the one that shows all the metabolism. It looks like this. And then we talk again about glucose to glycine, the idea of this metabolic pathway. Another metabolic pathway, fermentation, that we investigated with yogurt. And then we ask students, let me see if I can find it here, to select from this list one of these 15 different conditions that they are going to investigate. And the idea is that they investigate the condition, they do some research, and they find out what the enzyme is that's responsible for the condition, what the mutation is, and then also what the symptoms are, what effects the mutation has on the patient. And then they do a talk protocol with the bracket that I'll show you in a sec, and that will help them come down and come to a consensus as a class as to what baby Matthew's condition is. So we remind them about enzymes. We ask them to find a certain number of pieces of information about about the condition that they've been assigned. There is a shared slides template that you as teachers can make for them because you don't want multiple classes working on the same stuff. Otherwise, kids are going to find out and not have to do the work on their own. So I make a copy of the class slides for each section and I put it over onto the right-hand side here. And then they have the opportunity to investigate. So let me show you what that looks like. They see an image that looks like this, which is a cut out section of the larger poster. And you'll notice that on the right-hand side, we have a group of conditions. In this case, a small group, homocystinuria and maple syrup urine disease, both of which are defects in amino acid metabolism. And then, by the way, this diagram is all color-coded. So everything that's red is a protein. Carbohydrates are in a different color. Lipids are in a different color. What am I missing? Nucleic acids are in a different color. So, and I referred to this diagram a whole bunch of times all the way through the unit, so we're used to it by then. So a student group might choose this condition, maple syrup urine disease, to investigate. And so they look here where it says maple syrup urine disease, and it indicates that this enzyme number 1.2.4.4, which is it's the international naming system for proteins. That 1.2.4.4, if you were to type that into Google, it would actually spit back out this branched chain keto acid decarboxylase. So 1.2.4.4 has a very specific meaning, and that meaning is this name of an enzyme. And when students research this a little bit, they find out that there are these three particular branched chain amino acids, valine, isoleucine, and leucine, which cannot be broken down when this enzyme is defective. And it turns out that valine, isoleucine, and leucine, when present in the blood in high quantities, actually can cause things like seizures and brain damage under worst conditions. They also, when excreted, make the urine smell like maple syrup. Therefore, the name of the condition, maple syrup urine disease. So this is kind of a big eye-opener for students, because when we talk about the diaper in 2.1, they assume it has to do with his feces. But it's not having to do anything with his feces. It's actually his urine that has the odd odor. So I give you this example of what students might investigate. This, of course, is the condition that baby Matthew has, but they don't know that when they first select it. Some of them will select it just because it sounds funny. They think it's amusing to talk about excrement or, excretions. So, but you have to imagine that this is happening all around the room with all different kinds of conditions. And this is a hard diagram for students to try and figure out. There are a lot of big words on here. They get tripped up. They don't like to try to pronounce them because they are afraid they might look silly in front of their neighbor or their friends or whatever. So this is an example I like to talk to students about. You can do hard things. You can make statements about using scientific terminology. You can, there are lots of things that you can do that are difficult, and we're all going to work through it together. So then I wanted to show you the bracket. Where did that go? Oh, it's over here. So the bracket shows up like this. Once they've had some time to do their research and to write down all of the stuff they've learned about their condition that they are researching on their slide, we set them up in in opposing teams. And so here you have the top half of the bracket, and then the bottom half of the bracket. Whoever is number 15 is lucky, they get a bye in the first round. And then, and this is a slides, this is on slides, but you have the bracket also available in Google Sheets, and it has a drop-down menu associated with it. So you can keep track of it on the board, so that you don't actually, as the teacher, have to furiously type these long terms either. And if you're like me, invariably make spelling mistakes. And so then they work together with their partner and they have to argue their case. And this is why I wanted to talk about discourse strategies. But I give them these cards, and I physically have the cards. I actually have these cards often taped to the tables in my classroom so that they're always accessible and no student ever has to feel like they are, that it's an odd thing for them to use the card, because it's just part of what we do in science class, is that we have access to these supports. And so they present their case to the opposing team, whether they think that baby Matthew has this condition or not. And then they hear back from the other team. And then as a group of 4, 2 teams or 2 pairs of students working together at a table, they come to an agreement about which of the 2 conditions is the one that is more likely, and which of them should move on to the next round. And at that point, as they move then and they get transferred or moved out around in the room, and there are some teacher notes about how to effectively do this, or effectively how you might choose to do it, I should say. They argue for the same condition moving forward, but they split up. So eventually by the time you get to the championship round or the finals, you only have 2 conditions that are being argued, right? Because they're just kind of 2 finalists. All of the other conditions have been eliminated for one reason or another. So here are, and this is like the quick debate protocol that we have listed in here about how they would go about this. Sometimes you can, this is another situation, if you do it with shared slides and don't do the bracket, you can do something called multiple representations, where they have 4 different things that are showing up, and then they put a bingo chip or something next to the one that they think is the right one. But this doesn't work with the bracket. And then once we find out what the end result is, once we find out what baby Matthew's condition is, we talk about what's the relevance of this type of process. How is it that medical professionals come to an agreement about a diagnosis on a patient, especially when it's a complicated situation? Baby's only 6 days old. How are you sure that this is what's happening? What do they do? How do they work together as medical professionals to come to these conclusions? So having students have the opportunity to work together, to be expected to speak to each other, right? And here are things that they actually get, that they get out of it, right? If they practice their talking about their condition with their partner first, then if later on down the line in the process, they get separated from that partner in the bracket system, for example, or if you do a different type of jigsaw, they can feel a certain level of confidence in talking about that thing, even though it's a really complicated thing. For me, I really like the fact that I like the last bullet point, that this levels the power structure. I'm a co-investigator in this. We as a class are coming to some agreements about what we think the condition is. It's not like I'm holding back the knowledge from them. We are all working together towards the answer. So I really like that. That really helps my relationship with students, or my relationships that I am still in the process of building with them, right? Midway through the second semester or through the first semester, excuse me, when we're doing this. Jason, also, yeah, at the end, once it's determined that baby Matthew has maple syrup urine disease, I've never shown this in class, but I think I will do it this year. The video is quite compelling, because it talks about the family and how the family had to completely change the way that that all of them eat because of the limitations of their child. But the good news is that with the right kind of diet that's low in these branched chain amino acids, that children can live you can live a completely healthy life as long as you avoid certain kinds of foods with this condition. So I like just some basic, this is not from my classroom. My handwriting is not nearly as neat as that. But I think that it's important to have accountable talk norms in the classroom, right? Because especially in a patterns classroom where we're not necessarily doing a lot of lecture every day or even every week, the discussion between students and among students is really a major part of our responsibility to others, because students are transmitting knowledge that they and maybe a partner have. They're transmitting that knowledge to others in the room. I don't know every single thing, even as a teacher, I don't know every single thing about all of those 15 diseases. I rely upon the students to be the keepers of that knowledge and to share that through and to work through the thinking process. Another set of things that I like to use, and have another set of cards for this that I generally hand out when it's the right time to use them, are these ideas about talk moves, right? And that term talk moves has a lot of different meanings for teachers. It's like, how do we make talk moves that kind of ask a student to extend their thinking, for example? But I let students have these too, right? It gives them a little bit more power in the discussion process if they have access to these frames, so that they can encourage and help their colleagues get there. So of course, this last little bit has really been all about the design principles, right? We kind of have pulled back from talking too much about design principles in the webinars because there's so much content to talk about. But this is really about design principle 6, which is differentiation. How can we provide scaffolds for talking, right, that allow all students to be successful in those endeavors? All those different things. So we give them choices. We provide them, written sentence frames or spoken sentence frames, all those things. Really quickly here, we've talked about these along the way, but the different opportunities for assessment in this unit. And then we are going to give you, we're close to 5 minutes out. We'll ask for last minute questions, and then we do have an exit ticket that we would like you to do before you go. So we want to make sure that we give that to you before we leave. And let me pull that up. I'm just open it up to any questions, and I am going to pull a different link for the exit ticket. Yeah, I need to pull the exit ticket link and put it in the chat, but opening it up to questions at the moment.
Yeah, please
a
do. The
lot.
exit ticket is a grant thing.
Yeah, well, let me get them the link first.
Catherine, this one takes a long time. So I would say, for me, it takes a few months. As far as class periods go,
I think the last time I looked, it was like 18, but that's including time for assessment.
Yeah.
As opposed to, like, I'm finishing unit 1 in 15. So the two of those together is kind of long.
Yeah, usually first semester is 1 and 2 for me, and then I get into 3 second semester.
Okay, I finally got what I needed here. All right, I'm putting the exit ticket link into the chat. It is a requirement of the grant for us to supply ODE with exit ticket results. So if you could please fill out that exit ticket. It shouldn't take you more than the 3 minutes we have left. Oh, you're welcome. Thanks, Jasmine. Thank you, Sarah. Glad you were able to come. I should check see if we have anything in our webinar notes here. You're welcome, Kim. Thanks. Yeah, thank you. Bye. Okay, nothing on the parking lot.
Thank you.
Mhm.
Oh, you're welcome. Bye, Elena.
Bye.
Good to hear you. Bye.
So I have a quick question about the webinars
Sure.
and where they're stored.
Yeah, so let me cancel my sharing here. You don't need to see that.
No worries.
Let me pull up the PMSP website and see if I can help you navigate there.
That would be great because I think it took me to the YouTube channel. But what is shown here are the webinars from 2 years ago. So I just know my phone cut out. And so I missed a little bit of like a little chunk of time. But I also know that our PLC is not going to get to this unit for another probably 2 to 3 weeks. And so
Mhm.
it would be great since they weren't able to be here tonight, to be able to watch it together as
Yeah,
we're
great.
planning the unit.
Awesome. So let me look here. Now, to be completely honest with you, Molly, we don't have anything to do with that part of the process. This is not even our Zoom account. Like, this is a PMSP owned Zoom account. So the recording hangs out here and then someone at PMSP is supposed to deal with it. So I was not even aware that they were not going to the right location. So let me look here and see what I can find. Distance learning resources, professional development page. Here we go. Engineering. Okay, that's all physics. Where's the biology ones? Oh my gosh. Is biology even on here? October. Oh my gosh, it turned it off of upcoming. I wonder if that's interesting. Here, yeah, past. It's saying that today is past. So the question I think would be to find out whether or not they post the webinar recording on the same page where the information about this webinar was located. So I don't know the answer. We can ask and make sure that that information, Molly, makes it back to you.
That would
The-
be awesome if you were able to. And I mean, it's not one of those urgent things, but it would be lovely to have just because, you know,
For
others
sure.
weren't
I
able
mean,
to
and
make it.
the whole point, let me actually think, speaking of that, I can stop the recording now.